Hi everyone, we need to talk about osmosis. Now, osmosis is the spontaneous movement of water through a semipermeable membrane. A semipermeable membrane is a barrier that allows water and other small particles to pass across, but it doesn't let the large particles pass across. And osmosis is the diffusion of water and the diffusion of other things from low solute concentration to higher solute concentration is usually called diffusion, but we tend to use the terms diffusion and osmosis interchangeably these days. So that's just a little difference there. So if we look at the picture at the bottom, we can see that we've got a semipermeable membrane in the middle, and we've got on the left-hand side a region of high solute concentration, and on the right-hand side we've got a region of low solute concentration. And the large blue blobs, those represent some kind of large molecule. It could be a salt or something else. And the red molecules represent water or some other kind of very small solvent. And what we'll see is that the water molecules will spontaneously move from a region of low solute concentration to a region of high solute concentration. So what they're doing is they look at the other side and they say, oh, we need to seek and dilute. So they're going to rush over to the high solute concentration side and try to dilute it. And they'll try to dilute it until you have concentrations that are equal on both sides of the semipermeable membrane. And that's when we reach equilibrium. And at equilibrium, we don't have any net movement of our water from side to side. It stays stable, but that doesn't mean we have no movement. So if you get a few water molecules moving to this side at the same rate, you'll get water moving over onto the other side. So there's movement, there's just no net movement. Okay, so what happens is that this causes something called osmotic pressure. So the water is going to see a region of high solute concentration, and it's going to rush in there and try to dilute it because that's what water does. But then that's going to increase the volume here, so you can see it goes up that tube right there. And that is called osmotic pressure. So that's the pressure that prevents the flow of additional solvent into a solution on one side of a semipermeable membrane. So what we have is pressure over here that doesn't allow any more water to rush in there. And you can see a picture of that down here also. Now it says salt down here, but that doesn't just mean sodium chloride. That could be any kind of salt. So potassium chloride, potassium bromide, sodium nitrate, anything like that. And osmosis is really good for some species, and then some have a lot of trouble dealing with this, and over eons they have developed systems to take care of what osmosis causes, problems and whatnot. So with our trees, what happens is we have regions of high solute concentration inside the cell, and water's going to want to rush in there and dilute that. So when we have water doing that, it expands it inside, and so the cell membrane will expand until it hits the cell wall, because plant cells have a cell wall, and that makes them nice and firm. And then, as you can see in the picture over here, we can get nice tall trees and whatnot, and they don't have to have bones to keep them up because they take advantage of osmosis and the osmotic pressure that results from this. And again, we call this turgor. And if you think about when you don't water your plants, how they get all droopy, and then you water them and they stand back up, this is what's happening. The water is going into the cell and making the cells all nice and perky again, and then they stand up. What happens with animal cells is a little bit different because you don't have the cell wall. So it depends on whether you are in salt water or fresh water when you're a fish as to how your system has evolved to combat the problems of osmosis. So if we take a look at the fish on the left-hand side, this fish is in salt water, so there's a high concentration of salt on the outside of the fish's cells. So the water that's inside the cell wants to rush out into the salt water outside and dilute it. So what can happen in this situation is called crenation, when the cell shrinks. So the saltwater fish have to have mechanisms by which they keep the water content in their cells constant. And I'll leave that to biology and environmental studies to explain the systems that do that. Now, on the right-hand side, we have the freshwater fish. And in freshwater, you have low salt content. And so that means that there's a higher content of salt and other particles, and that makes water want to rush into the cell. And the problem is that that can blow up the cell, and that's called lysis. So remember, we don't have a cell wall around our cell membrane in animal cells. So if we have too much water, it'll just split the cell open. So freshwater fish have developed mechanisms by which they can get the water pumped out of their body that they need to pump out in order to keep their cells from bursting. So those are the issues that the two different fish face. So in this slide, we've got hypotonic and hypertonic solutions. Now, a hypotonic solution, this is a situation in which the concentration of the solute is higher inside the cell than in the surrounding solution. So if we look over at the picture, we've got 20% salt, again, whatever the salt is, on the inside of the cell, and 10% on the outside of the cell. So the water that's on the outside of the cell is going to want to rush in and dilute the situation on the inside of the cell, because that's what water does. It's like seek and dilute. And if we don't adjust for this, we're going to get cell lysis. If you've got a cell wall around it, you'll get turgor, and that's okay. But if you don't, you're going to get cell lysis. And this is the problem that our freshwater fish face. Now, down below, we have a hypertonic solution, which means we have a higher salt content on the outside. So we have a higher solute concentration in the solution on the outside of the cell. And so the water on the inside of the cell wants to rush out here and dilute the solution on the outside of the cell. So this causes cell shrinkage, which we already said on the other slide was called crenation. And this is the problem that our saltwater fish face. So they have to have a mechanism to keep the water inside the cells. If we have equal salt concentrations on each side of the cell. This is called isotonic. So an isotonic solution is also called a physiological solution. So the water isn't going to want to rush to either side of the cell to dilute the salt concentration because it's equal on both sides. So there's no net movement of water. You don't have water moving to one side of the cell or the other in greater amounts. Now, again, that doesn't mean that water doesn't move at all. We will still have a few water molecules moving through the cell membrane out into the solution out there, but at the same rate, we'll have a few water molecules moving in. So we keep the concentrations constant on each side. And that brings us to the end of chapter 8, my friends. Yay!